Hello and welcome to Molecular Biology. My name is Martin. I'm going to be bringing you through these 6 topics of this subject. Today we're starting with topic 1 on Introduction to Genetics. Now let's go through some conventions first. There are some texts in this stack of slides which are bolded. These are key terms and concepts which are part of the learning outcomes of the subject. So in other words, these are the things that you probably have to pay extra attention to. There are a couple of slides which are labeled FYI at the bottom. These slides are for your information only. You will not be tested on the contents of these slides. And finally, the handouts that you're given is actually missing some information. So you need to actually watch the lecture videos so that you can actually fill in the blanks. Alright, let's go into topic 1, Introduction to Genetics. After the first topic, you should be able to apply Mendel's laws of inheritance to the concept of genes as functional units of heredity and variation. There are 3 parts in this topic. The first part is on Mendelian inheritance. The second part is on how to construct the Punnett square. And lastly, we'll go through some non-Mendelian inheritance as well. Part 1 on Mendelian inheritance. These are the learning outcomes of this part. You need to be able to define the terms gene locus, allele, homozygous and heterozygous, genotype versus phenotype, dominant versus recessive, progeny, F1 and F2 generation. You should be able to describe the concept of genes as functional units of heredity and variation. And lastly, you need to be able to explain Mendel's laws of inheritance. In this topic, we're going to look at Mendelian inheritance. And these are some of the questions that people are asking themselves over the history. Why do children resemble their parents or their grandparents? And why do children of the same parents resemble each other? And Mendel was the one person that actually found some answer to that. And these are some of the key concepts that we're going to go through in this part of the topic 1. The first one is on genes. Genes are the basic units of biological information. They are the regions in the DNA that specifies a particular protein or an RNA. Next, we have locus. Locus is basically the designated location on a chromosome. Oftentimes, the word gene and locus are used interchangeably. Next, we have alleles. Alleles are basically different forms of a single gene. Next is genotypes. Genotypes are the alleles present in an individual. Traits are specific characters. Traits are specific characteristics of an individual. These can be determined by genes or by environmental factors or a combination of both. And we also call traits phenotypes. And lastly, heredity is the passing of genes from one generation, which is usually termed parent, to the next generation, which is usually termed progeny or filial. Now, why is this classic genetics theory called Mendelian genetics? It's because it's actually discovered by this person, Gregor Mendel, who's a monk in Austria. He discovered the basic principles of genetics while experimenting on very clear-cut alternative traits in pea plants. From there, he discovered the patterns of inheritance, which explains the similarities and probably differences of different individuals from the same family. If you want to know a little bit more about this, you can watch this YouTube video. I'll provide the link in your slides as well, so you can take a look at it. The experiment that Mendel did was basically cross-fertilizing pea plants in order to actually observe the phenotypes that actually show up in the progeny. So what he did was basically cross-fertilizing the pollen from one pea plant onto the other pea plant's stigma. In picture A here, this is the pea plant, Pisum sativum, with the white flowers that you see here. Picture B is anatomy of the pea flowers. And you can see it's got a green color stigma in the middle, okay, and it's got the anthers, which produce the pollen at the outside of it. Now, typically, pea flowers actually self-fertilizes because the anthers are actually in pretty close proximity to the stigma. But what Mendel did was actually cross-fertilizing the flowers by actually first removing the anthers from the flowers so that the flowers will not be able to self-fertilize. So basically, he will cut this away. Okay, cut this away so that the flowers won't be able to self-fertilize. And after that, he takes the pollen from this flower and using a brush, you can see from here, he's got a brush here. Okay, so he's using this brush to actually collect some of the pollen from another flower and to transfer the pollen onto the stigma so that this white color flower, as you can see here, this white color flower is actually receiving the pollen from this purple color flower. So that's how Mendel actually performs a cross-fertilization. And the result of that is the seeds, or peas, okay, in this case, the peas will actually be fertilized by the pollen of a different plant, therefore carrying hopefully the genes from that other plant. And through this cross-fertilization process, Mendel was able to actually produce hybrid pea plants from pure breeds. Hybrid meaning these pea plants actually have pollens coming from a different plant, but they are hybrids. Okay. He did actually notice that the first filial generation, the first generation that comes from the parents of the hybrids actually similar. They actually appear quite similar to one of its parents. Okay. So if you take a look at this example on the right, we have three different kinds of phenotypes that we actually to look at. The first one is the seed color, second one is the seed shape, the third one is the flower color. So if you cross a plant with yellow seed with one with green seed, and you can see that the first filial generation, which is this one, has yellow seeds. Similarly, you cross a plant with brown seed with one with wrinkled seed, you end up with a filial one generation with only brown seeds. And also, crossing purple flower with white flower give you purple flower in the first filial generation. So as you can see, all these first filial generation have a characteristics which is similar, or the same actually, as one of its parents. So Mendel calls these characteristics which actually appear in the first generation, the first F1 generation hybrids, as dominant. And the antagonist characteristic, which remains hidden in the first generation, is then called a recessive characteristic. After getting the first F1 generation, Mendel then went on to conduct a monohybrid cross, where the F1 generation, in this case we're just taking these yellow seeds, this F1 generation was allowed to self-fertilize. So in other words, he will not remove the anthers from these flowers so that they will be able to actually spread their pollens onto the stigma of these flowers. And then he will allow this second generation of peas to actually grow. And he noticed that in the second filial generation, phenotype that went missing in the first generation, which is green peas. Green peas were missing in the first generation, but it appears in the second filial generation. And he also noticed that even though green pea phenotype did reappear, the amount of green peas is actually a lot less than the yellow peas. In fact, he actually counted the number of the peas that he got, and he had about 6,000 yellow peas and only about 2,000 green peas. So the ratio is around 3 to 1. Now we will come back to the ratio in a moment, but for the moment being, let's just try to remember that in the second filial generation, there is a possible phenotype ratio of 3 to 1 of the dominant and recessive phenotypes. So from these experiments, Mendel came up with his first law of inheritance, which is the Mendel's law of dominance. The Mendel's law of dominance states that there are two possible alleles for each gene. One of them is dominant, which is usually denoted in capital letters, and the other allele rather, is recessive, which is usually denoted in lowercase letters. Each individual has two copies of a gene, which are basic combinations of these two alleles. And the individual will always display the dominant phenotype when the dominant allele is present, whereas the recessive phenotype will only display when both copies of the genes are recessive. And because of all these, hybrids, which are individuals with a copy of the dominant allele and a copy of the recessive allele, will actually display the dominant phenotype. And this is Mendel's law of dominance. Based on Mendel's law of dominance, we can see that there are three possible genotypes which can arise from one gene. The first genotype is called homozygous dominant, where this individual will have two copies of the dominant allele, which in this case is our first example here. This is a homozygous dominant individual with two copies of dominant allele. The second possible scenario is called heterozygous. An heterozygous individual will have one copy of the dominant allele and one copy of the recessive allele in this genotype, which is this one. So you can see there is a dominant allele plus a recessive allele in this individual. And the third scenario is called homozygous recessive, which are individuals who have two copies of recessive allele in its genes, which is the third situation here. You can see both alleles that this individual contains are recessive. And you can see that both the homozygous dominant individual and the heterozygous individuals both display the dominant phenotype. Only the homozygous recessive individual will actually display the recessive phenotype. Based on his findings, Mendel also went on to propose his second law of inheritance, which is Mendel's law of segregation. By Mendel's law of segregation, he says that each of the inherited traits that we can see is actually defined by two gene copies. In other words, every individual will actually have two alleles of the same gene. Of these two alleles, when the individual is actually producing gametes, which are sex cells, which you guys probably have learned in CPB, the egg and sperm cells, only one of these two gene copies is going to be randomly distributed into each gamete. So each sperm or egg cell will contain one copy of the two genes. And when the gametes come together during fertilization process, the offspring then inherits only one copy of the gene from each of the parents. So there are two processes which are discussed in Mendel's law of segregation. The first process is the process of producing gametes, which is shown in this first picture. So as you can see, we have two pea plants. One is the yellow one, which, as you can see, has the genotype of dominant Y and dominant Y. Right. So this is actually a homozygous dominant individual. This is coming from a pure breeding stock. On the other hand, we have this green pea, which contains two recessive copies of the Y gene, coming from a pure breeding stock again, and this is homozygously recessive. So when these two pea plants grow into these two peas grow into a plant, they will be able to actually produce sperm or egg cells. In each of the sperm or egg cells, they will actually contain only one copy of each of the genes. So for the yellow pea plant, it's able to produce only gametes with the dominant allele, whereas for the green pea, the one that's homozygously recessive, is only able to produce gametes which contain the recessive allele of the gene. Now, when the gametes of these two plants come together, which is the situation that we're seeing here, if we were to fertilize, the plants using this kind of combination, we will have a zygote. In this case, this one that we have here, which is actually a hybrid. Okay, it has one copy of dominant gene, dominant allele, and one copy of the recessive allele. And this zygote will go on to be developed into a full seed with a genotype, which is a heterozygous genotype. But of course, in terms of the phenotype, it's going to actually demonstrate the dominant phenotype, which is yellow peas. So how did the segregation happen? This is basically the cellular mechanism for the Mendel's laws of segregation. You probably have learned about this in CBB, where we talk about meiosis. Okay. So during the meiosis process, the chromosomes will actually replicate, and cell will actually progress into the metaphase of meiosis 1. And during that process, the homologous chromosomes will actually segregate into the separate cells. So as you can see here, these two chromosomes will actually segregate into two different cells, therefore segregating the two alleles that this individual is actually carrying. And then after that, the cystochromatids will segregate into the separate cells during the anaphase of meiosis 2. So in these two haploid cells, each one of them will actually contain or carry one of the possible alleles of this gene. Mendel's third law of inheritance is that of independent assortment. In Mendel's law of independent assortment, it says that the alleles of two or more different genes will actually get sorted into gametes independently of one another. So in other words, the gametes will actually contain all possible combinations of the alleles. Let's take a look at an example that's given here. So in this parent generation that we have, this parent generation has two genes that we're looking at, both the Y gene and the R gene. For this parent cell, it's actually heterozygous on both of the genes. So we have the dominant Y recessive Y gene, as well as the dominant and recessive R genes in this parent cell. So during gamete formation, the Y and R genes are actually assorted independently into the gametes. So in other words, there is a 2-in-1 chance that the gamete is going to get a recessive or a dominant Y gene. And there's also a 50% chance that it's getting a dominant or recessive R gene. So therefore, it's actually able to generate, so for this parent cell that we have, it's actually able to generate 4 different kinds of gametes. These 4 different kinds of gametes. Okay. So the gametes are either carrying the dominant Y gene, which are the first 2, or the recessive Y gene, the bottom 2, the dominant R gene, which are the first one and third one, or a recessive R gene, which are the second one and the fourth one. And for each of these 4 possible gametes, the likelihood of them occurring is equal. Okay, so there's a 25% or 1/4 possibility that these gametes are actually occurring. So this is Mendel's law of independent assortment. Now, one thing that you have to note, rather, is that the law of independent assortment applies only when the genes are actually not linked. So in other words, they are not on the same chromosome. If the genes are on the same chromosome, they won't actually be sorted independently. Again, let's take a look at the cellular mechanisms behind Mendel's law of independent assortment. We're starting with the cell, which is a heterozygous diploid cell, which is undergoing meiosis. So in this case, if you look at this cell here, it's actually heterozygous for both the Y and R genes. Okay, so as you can see, it's carrying a recessive and dominant allele of Y, as well as a recessive and a dominant allele of R. Now, when these cells undergo meiosis, during the first metaphase, the alignment of the 2 homologs can actually occur in more than one way. Okay, so as you can see, this is actually the first way that it can actually occur, the one on the left. So as you can see, the recessive Y chromosome is being sort of aligned with the dominant the dominant R chromosome. Whereas in this other situation on the right, the dominant Y allele is being aligned with the dominant R allele. So these are 2 different possible situations. Of course, if you're looking at more genes and more locuses, basically, you'll have a lot more different kinds of combinations that can happen. And when these cells go on to finish their first step of meiosis, you will have 2 different kinds of gametes that will actually form after this process. This first set of gametes, you can see that the dominant R and recessive Y are assorted into one kind of gamete. Dominant Y and recessive R are also assorted into one kind of gamete. So this is one situation that can happen. In a second situation, the dominant Y and dominant R cells are then assorted into one kind of gamete, and the recessive Y and R alleles are assorted into one kind of gamete. So from one cell that we have here, which is heterozygous on both the Y and R gene, we end up with 4 different possible gametes that it can actually generate. The ones with the dominant R and recessive Y, dominant Y and recessive R, dominant R and dominant Y, and recessive R and recessive Y. And that is basically how Mendel's independent assortment actually takes place. And with that, we have come to the end of part 1. In the next part, part 2, we'll be looking at how to use the 3 Mendel's laws of inheritance to actually construct Punnett squares. Punnett squares are a very useful tool for geneticists to actually predict frequencies of genotypes and phenotypes in future generations. I'll see you next then. Welcome back. In part 2 of topic 1, we're looking at how to construct Punnett squares. These are the two learning outcomes of part 2. First one is to construct a Punnett square to display the genetic crosses. And second one is to interpret various gametic combinations and the results of various mono and dihybrid crosses. This is just a quick recap of the three Mendel's laws of inheritance: Mendel's law of dominance, law of segregation, and law of individual assortment. And based on these three Mendel's laws of inheritance, we can construct Punnett squares to predict results of a cross of any two individuals. Punnett squares are actually visual tools to help us summarize a cross. And in the next couple of slides, I'm going to actually walk you through the construction of the Punnett squares. Let's start with a situation called a monohybrid cross. A monohybrid cross is the hybrid of two individuals which are homozygous for different alleles of one gene locus. So in this example that we're giving you, we are crossing two individuals, okay, pea plants, with yellow and green peas. So for the yellow pea that we have here, this pea plant is actually homozygous for the dominant Y gene, and the green one is homozygous for the recessive Y gene. We're going to use Punnett square to demonstrate a monohybrid cross using these two pea plants. Now, to construct a Punnett square, the first step we need to do is to write down the genotypes of both of the parents. For the female parent here, as you can see from the picture, is actually a yellow pea. Right? So for the female parent, the genotype has to be homozygous dominant of the Y gene. On the other hand, for the male parent, it has to be homozygous recessive of the Y gene. The next step is to write down the possible gametes that each of the parents can make. For the female parent, again, because it's homozygous dominant, it's only able to produce the dominant Y gametes. Whereas for the male parent, it is again only able to produce the recessive Y gamete. Now, based on Mendel's law of segregation, for the F1 generation, the only possible genotype is heterozygous on the Y gene. So as you can see from here, the F1 generation, they are both heterozygous on the Y gene, and the phenotype is then yellow peas. Moving on, in a monohybrid cross, the F1 generation is actually allowed to self-cross. So these two pea plants that we are able to generate in the F1 generation, they will actually go on to self-cross. And both the male and female parents now have two possible gametes. Let's take a look at the female one first. The female parent, through its eggs, produces either the dominant Y gamete or the recessive Y gamete. Because the female gametes are actually written on the left of the Punnett square. For the male parent as well, it's able to produce one dominant Y gamete and one recessive Y allele gamete. So when you are creating an empty Punnett square, you are going to actually draw a 2x2 square, which is basically this one. Okay, you're going to draw a 2x2 square, and you're going to write the possible male gametes on the top, and you're going to write the possible female gametes on the left. The next step in constructing a Punnett square is to fill in the possible genotypes into the empty spaces. Now, starting from the upper left one, you can see that this individual received a dominant Y from the father and a dominant Y from the mother. So this individual is then homozygous dominant of the Y gene. Let's move on to the right. You can see that this individual received a recessive Y from the father and a dominant Y from the mother. So this guy is heterozygous on the Y gene. The same situation happens for this fella as well, heterozygous on the Y gene. And lastly, we have the lower right corner one, which receives a recessive Y from the father and a recessive Y from the mother. This one is homozygous recessive on the Y gene. So based on this Punnett square, we are able to actually get 3 different kinds of genotypes: homozygous dominant, heterozygous, and homozygous recessive. And the genotype ratio of these 3 genotypes is 1 to 2 to 1. 1 homozygous dominant, 2 heterozygous, 1 homozygous recessive. When it comes to the phenotypes, the phenotype ratio is actually 3:1. Okay, where we have the tree, which is yellow peas. This tree will contain yellow peas. And we have this one on the lower right corner, which has green pea. Does the 3:1 ratio look familiar? We actually saw this 3:1 ratio earlier on when we were discussing Mendel's experiments. So when Mendel did this experiment, he actually received, in the filial 2 generation, about 6,000 yellow peas and 2,000 green peas. And the ratio here is exactly 3:1. So before the era of Punnett squares, Mendel already kind of has an idea through experiment what the phenotypic ratio is going to be in a second filial generation if you start from pure breeds of the yellow and green peas. Next, let's take a look at a slightly more complicated scenario: a dihybrid cross. A dihybrid cross is the hybrid of 2 individuals which are homozygous for different alleles of 2 gene locus. In this example that we're giving you, we are crossing pea plants with yellow round and green wrinkled peas. So the yellow round pea plant is homozygous for the dominant Y and R genes, which is the one on the left here. Okay, you can see it's homozygous on both the Y and R gene, homozygous dominant. The pea plant on the right has green wrinkled peas. Okay, so he's homozygous for the recessive Y and R genes. Again, similar to a monohybrid cross, the first step into constructing a Punnett square is to write down the genotypes of both of the parents. So again, the parent on the left, which assuming it's the female parent, is homozygous dominant of the Y and R gene, and the male parent on the right is homozygous recessive of the Y and R genes. Next, let's write down the possible gametes that each of the parents can make. So for the female parent, the one on the left is only able to create gametes which contain the dominant Y and dominant R genes. And for the male parent, the gametes that it can produce is only recessive 1 and recessive R. Now, based on these 2 gametes, we can already know that the F1 generation will be all identical, and they will carry, they will be actually heterozygous on both the Y and R genes. Just change the color here. So this generation, heterozygous on both the Y and R gene. And the phenotype you're going to observe is yellow round piece, which is basically the dominant phenotype for both the Y and R genes. Moving on, the F1 generation is then again allowed to self-cross, and based on what we did previously, well, with monohybrid cross, we are then going to actually construct a empty Punnett square, which is this one, a 4x4 table. And we are going to write down the possible gametes that the F1 generation is able to generate on the left and on the top of this table. So let's start with the female parent in the F1 generation. So for this particular genotype, based on Mendel's law of independent assortment, we can see that it's actually able to generate 4 kinds of possible gametes: dominant Y and dominant R, dominant Y, recessive R, recessive Y and dominant R, and lastly recessive Y and recessive R. The same situation happens with the male parent as well. We have these 4 possible gametes that the male is able to produce. And the likelihood of each of these 4 situations is equal. So each one has a 25% chance of occurring. And based on this, we can then create this empty Punnett square, 4x4 Punnett square for the next step. In the next step of constructing Punnett square, we will then go on to fill in the possible genotypes. Now, similar to the previous one that we just did, we're going to match the male gamete, the female gamete, and then we're going to write down the possible genotype of that particular individual in that intersection. So I'm not going to go through each one of them because there are 16 of them, and you can actually go ahead and write down the genotypes of all the 16 individuals that's in the Punnett square. So there are altogether 9 possible genotypes that you can generate. Homozygous dominant for Y and R gene, homozygous dominant Y and heterozygous on R, heterozygous on Y, homozygous dominant on R, heterozygous for both Y and R gene, homozygous dominant on R, homozygous recessive on R, heterozygous on Y, homozygous recessive on R, homozygous recessive on Y and homozygous dominant on R, recessive homozygous recessive on Y and heterozygous on R, and finally homozygous recessive on Y and R both. And the genotypic ratio is 1 to 2 to 2 to 4 to 1 to 2 and 1 to 2 to 1. Now it's quite a mouthful. You can get all these ratios from the Punnett square. Okay, you just basically need to count the number of occurrences that each of these genotypes actually have. In terms of the phenotype ratio, there are 4 possible phenotypes that you can observe. Yellow round P, which is the dominant phenotype for both genes. Yellow wrinkle P, so dominant on the color gene and recessive on the shape gene. Green round P, dominant recessive on the color and dominant on the shape. And finally, green wrinkle P, which is recessive for both of the color and shape genes. And the phenotypic ratio is 9 to 3 to 3 to 1. Again, it's a fairly typical phenotypic ratio for dihybrid crosses. And Mendel did actually get the same kind of result from his experiments. As you can see, from his experiments, the yellow round, green round, yellow wrinkled, and green wrinkled phenotypes have a phenotypic ratio of 9 to 3 to 3 to 1, which is exactly what we predicted with our Punnett square. And if you zoom down to the individual phenotypes, as you can see here, the yellow to green is 3 to 1, and round to wrinkled is also a 3 to 1 ratio. So this is how you can actually use Punnett squares to predict the genotypic and phenotypic ratios for different combinations of genes. Next, let's take a look at a special kind of crossing, which is called a test cross. Test crosses are useful in identifying the genotype of an individual which displays the dominant phenotype. When an individual is displaying the dominant phenotype, it's very hard to actually judge based on the phenotype alone what is the possible or underlying genotype behind this phenotype, because the individual may be homozygous dominant for that gene, or it can be a heterozygous for that gene. And a test cross is there to help you identify the underlying genotype. When doing the test cross, this individual is crossed with another one which is known to be homozygous recessive on that particular gene of interest. And based on the phenotypes of the F1 generation, we will then be able to actually identify the genotype of the parent. Let's take a look at the 4 examples that we have on the right. In this particular example, we have 4 different individuals with different genotypes. This first one is homozygous dominant on both the Y and R gene. The second one, homozygous on Y, homozygous dominant on Y, and heterozygous on R. The third one, heterozygous on Y and homozygous dominant on R. And the fourth one, heterozygous on both the Y and R genes. Now, these 4 individuals have different genotypes, but on the phenotype front, you can see that they will actually all display the dominant phenotype. And this is the P, color, and shape gene. So all these 4 will actually be yellow round P's. So how do you identify if you encounter a yellow round P? How do you identify the genotype of this particular P plant? What you can do is actually conduct a cross of this plant with another plant which you know is homozygous recessive. Okay, in this case, this is the other plant that we are actually crossing it with. See, these are all homozygous recessive plants. Now, when you cross these 2 plants, 4 possible scenarios may happen. Let's take a look at the first one. This parent plant is able to produce only one kind of gamete, which is dominant Y and dominant R. Now, when you cross this with a another plant which is homozygous recessive on both genes, you only have one possible phenotype in the F1 generation, which is yellow round P's. Okay, so if let's say after a test cross you get 100% yellow round P's, you will know that the parent generation is actually homozygous dominant for the Y and R genes. The second situation, the individual is heterozygous on the R gene. So this individual is then able to produce 2 kinds of gametes, these 2 gametes. And then when you mix this with the recessive Y and R, you'll get 2 possible phenotypes in the F1 generation. Okay, yellow round and yellow wrinkled. And since they are all yellow, you can you can be sure that the Y gene should be dominant in the parent generation, and the R gene, because there is actually a 1-to-1 ratio of the round and wrinkled peas, you can know that the R gene is heterozygous in the parent generation. Okay. In the third situation, the parent again is able to generate two kinds of gametes, and then you have these two situations here: yellow round peas and green round peas in a 1-to-1 ratio. There's only one kind of shape that you have, so you can be very sure that the parent generation has only the dominant R here. But because there are two different colors in the F1 generation, this parent has to be heterozygous on the Y gene. The last situation, you have four kinds of phenotypes that appear in the F1 generation, right? You have yellow round, yellow wrinkled, green round, and green wrinkled. So based on this phenotypic ratio, 1-to-1-to-1-to-1, you can be very sure that the parent generation is able to produce four kinds of gametes. So the parent generation has to be heterozygous on both the Y and R genes. And this is how you actually use test cross to identify the genotype of the parent generation, even though the parent generation displays a dominant phenotype. This is what we have just discussed, so you can take a look at the explanation on the slides. Okay, so that brings us to the end of part 2, where we discussed how you can construct Punnett squares and how you can use them to predict phenotypes and genotypes of future generations. In the next part of topic 1, we'll look at non-Mendelian inheritance. We'll look at a couple of situations where the inheritance pattern doesn't necessarily follow the Mendelian rules. I'll see you there. Welcome back to part 3 of topic 1. We'll be looking at non-Mendelian inheritance in this part. And the learning outcome is for you to identify the different types of inheritance, such as sex-linked inheritance, codominance, and incomplete dominance. Now, in the first two parts of topic 1, we talked about the different Mendelian inheritance patterns. But there are also a lot of characteristics or phenotypes that don't actually fall neatly into the two opposing categories that Mendel actually proposed. Some of the characteristics actually appear to have more than two phenotypes originating from the same gene. Some of the characteristics appear to have more than one dominant phenotype. And some characteristics appear to be gender-specific. And there are also instances where characteristics actually appear to be affected by multiple genes. So the Mendel's laws are then extended for single and multiple gene inheritance. Now let's look at single gene non-Mendelian inheritance, which we'll be looking at different kinds of dominance patterns. There are three different kinds of dominance relationships which geneticists have already discovered. The first kind is the complete dominance. This is what Mendel actually experimented on in his experiments. So this is the type of dominance relationship where the hybrids will only display the dominant phenotype. So as you can see from the picture here, the first row is complete dominance. So if you let's just assume that the A1 gene is the dominant allele and A2 is the recessive allele. So a hybrid coming from a pure breed of A1 and A2 will only display the phenotype of A1, which is the dominant allele. So that's complete dominance. the one that we have always been talking about. The second one is incomplete dominance. In the case of incomplete dominance, the hybrids will actually display the phenotype which is unlike either of the dominant or recessive phenotypes. So this is the incomplete dominance situation. So again, we have the cross between a homozygous dominant and homozygous recessive individual, and the hybrid, which is this one, is not like either of the parents. Now, to make things even more interesting, there's also a thing called codominance. Now, in the case of codominance, this gene may actually have multiple possible dominant alleles. So in this particular case that we have in this picture here, both A1 and A2 are actually dominant alleles. And when you have hybrids of A1 and A2, these individuals actually display both the A1 and A2 phenotypes together. So they are actually displayed at the same time. So that is what codominant situations are like. Let's take a look at a example of incomplete dominance. In this example, we're looking at snapdragon flowers. The color of these flowers is actually controlled by one gene. So the dominant phenotype is red flowers, and the recessive phenotype is actually white flowers. So in this monohybrid cross that we have here, the red color flowers are crossed with white color flowers. So red color flowers are homozygous dominant, white color flowers homozygous recessive. And the F1 generation, which are hybrids, they are actually all pink. Okay, and the pink flowers are unlike any of its parents. Now, one of the possible reasons that people find about the color of the snapdragon flowers is that it actually the dominant phenotype actually expresses an enzyme which produces the red color pigment. So for the individuals which are homozygous dominant, it actually produces enough red pigment for the color to actually become red. Now for the heterozygous ones, which are here, these individuals produce about only half of the enzyme that the red color flowers are actually producing. So it's actually only able to produce half of the amount of red color pigment as well. So these flowers would then show a lighter shade of red, which is pink color. So that's potentially the reason why the flowers are actually pink. Now, if you look at the self-crossing of the F1 generation, you can see that you can still draw a Punnett square, and the incomplete dominance will still actually follow a Mendelian distribution of genotypes, which is 1 to 2 to 1, where the 2 is actually pink color flowers. So you can actually still use a Punnett square to predict the phenotypes in the situation of a incomplete dominance. Next, let's take a look at codominance. Codominance is a pattern of inheritance where both the alleles of a trait are expressed in a heterozygous condition, and both alleles are actually equally dominant and contribute to the phenotype. One of the very classic examples of codominance is the ABO blood type in humans. So for humans, there are 4 different possible blood types, right? A, B, AB, and O. Now, these 4 different blood types are actually governed by one gene, and there are three different possible alleles in this blood type gene. There are two dominant alleles, IA and IB, and there's one recessive allele, I. So the different combinations of these alleles will contribute to the different blood types in the humans. So blood type A is usually either a homozygous dominant individual on IA, or it could also be a heterozygous of IA and recessive I. For blood type B, again, we have two possible situations. Homozygous on IB, or heterozygous on IB and recessive I. And then we have the O blood type, which is homozygous on the recessive I gene. Now, the codominance happens in blood type AB, where these individuals actually have a copy of dominant IA and a copy of dominant IB. And these different genes are actually responsible for the sugar molecules which are presented on the surface of red blood cells, as you can see from this picture. People with blood type A or B will be presenting only A or B sugars on the surface of their white blood cells, the red blood cells, and people with blood type AB will be presenting both A and B sugars. Now, imagine if a person is carrying blood type A, so maybe a person is of blood type A, so basically the red blood cells in this person has all the A sugars on the surface. If you were to take this person's blood and transfuse the plasma into a person with blood type AB, the person will still survive because this person's red blood cells actually contain the A sugars as well. But if you put this person's blood into blood type B, it won't work because people with blood type B, the red blood cells don't actually carry the A sugars, so this will actually cause the blood cells to clump up and eventually lead to death of the person. So A to AB works, but A to B, it won't work. You'll learn more about this later on in blood banking, but this is just a very simplified version of the ABO blood type. But just remember, ABO blood type is a very classic example of codominance, and these are what's contributing to the different blood types in humans. The third example of non-Mendelian inheritance we're going to be looking at is sex-linked inheritance. Now, as the name suggests, we're talking about inheritance of genes which are on the sex chromosome. As you may have learned previously, for a lot of organisms, the sex chromosome determines the gender of the particular organism. So including humans also, our sex chromosomes determine the gender of us. So for genes which are on the sex chromosome, they will follow the chromosome to the gametes. The interesting thing about sex chromosome is that for females, they actually carry two X chromosomes. Whereas for males, they only carry one X chromosome. So therein brings in a lot of issues or a lot of problem. So the first problem is that for defects on the X chromosome or recessive alleles on the X chromosome, for females, they need to have two copies of that particular recessive gene, that recessive allele, in order to actually display the recessive phenotype. Whereas for males, as long as they have one copy of the recessive allele, they will actually display the recessive phenotype already. So in other words, for males, they're actually more susceptible to X-linked genetic diseases because they only have one copy of X chromosome. Another situation is that sometimes these genetic problem, or that there's this mutations, may also happen on the Y chromosome. And in that situation, only the males will actually get those alleles. Okay, and you will actually see that if you see this on the family tree, every single male in this family is actually getting a particular allele, and that is because this allele is actually inherited on the Y chromosome. So that's what sex-linked inheritance is like. Let's take a look at the example that we have here. We're looking at the gene that's determining the fruit fly eye color. So there are two. So this gene is actually on the X chromosome. So there are two alleles. One is the XW plus, and this is the dominant allele. And the other one is the XW, and this is the recessive allele. Now let's take a look at this cross here. The female individual here is actually a heterozygous on the W gene. So you can see it has one copy of the dominant W gene and one copy of the recessive W gene. And for the male individual here, it actually has the dominant W gene as well. So both of these fruit flies have red colored eyes. Now when they mate and they make their first generation of children, it's filial one. So if you look at the filial one generation, you have these two, which are females, and these two, which are males. Now the females will all have red color eyes because they may be homozygous on the W gene or be heterozygous on the W gene, regardless, because they carry two copies of the W gene, and one of them is definitely gonna be dominant allele. So these two, so all the females that's coming from this cross is going to carry the dominant W gene regardless. So they will be having red eyes. Whereas for the males here, you have a 50% chance where the males are actually getting the dominant W gene, and a 50% chance where they are getting the recessive W gene. So there is a 50% chance that the males actually have they will actually display the recessive phenotype because there's only one copy of the dominant of the recessive gene, and there is actually no dominant gene present in these males. So these males will actually display the recessive phenotype, which is white eyes. So in this particular example that we have here, only the males will actually have white eyes. Now this is a very, very similar example as a human genetic disease, which is color blindness. Okay, again, color blindness is a X-linked recessive disease. Very similar in inheritance pattern to this. So you can expect more males to be actually colorblind than females. In fact, I think about 7% of more males are actually colorblind, red and green colorblind, and only about 0.5% of females are red and green colorblind. And that is actually quite prevalent in here. Okay, so that's all of part 3, and that's the end of topic 1. I'll see you guys in the next topic.